All right, so this is going to be the last video on structures, properties, and functions of macromolecules. And this will probably be the shortest one because it's about nucleic acids. And the reason that it's the shortest one is that I'm not going to be completely exhaustive with it right now. Because later on, when we start genetics, we are going to have a whole unit about DNA and RNA. And we can have a bunch of time to get in a lot more specific detail about it then. I just want to provide the overview of the material now so you have enough for the purpose of this unit. So, as far as nucleic acids go, their main function is to serve as genetic material. One way they do that is by storing information, genetic information in the form of genes. All right, that's like DNA's job to store hereditary information in genes, to act as a blueprint for building proteins, that DNA RNA protein pathway that we will talk about later in genetics in protein synthesis. So in the case of DNA, it stores genetic information. And in the case of RNA, it transfers that information into building new cells or into building new proteins. So storing and transferring genetic information into new cells, into proteins, that's the job of nucleic acids. And we will only talk about DNA and RNA as nucleic acids in AP biology. So here are the two that I just mentioned, DNA and RNA. I kind of switched them around, but we're going to talk about DNA first. DNA is short for deoxyribonucleic acid. All right. And like the picture shows on the bottom, it is a double helix. It has two strands. One strand here, two strands here. DNA is double-stranded. It contains deoxyribose. OSE, that's a sugar. Remember, that's a carbohydrate. And DNA contains the base, the nitrogenous base, thymine. On the other hand, RNA, or ribonucleic acid, is a single helix. It only has one strand. Only one strand here. It contains ribose sugar instead of deoxyribose sugar, and it has uracil in it instead of thymine. We'll talk about the differences between RNA and DNA more when we get into genetics, but these are the ones that are important to remember for macromolecule structure, function, and property right now. The monomers of all nucleic acids, DNA and RNA, are called nucleotides. Nucleotide is the building block, and we'll see that on the next slide. So here I have a nucleotide. I can have a DNA nucleotide and an RNA nucleotide, but they all contain three parts. Some sort of nitrogen base. That means it has nitrogen in it. All right? A, T, C, and G. Adenine, thymine, cytosine, and guanine. All right, a nitrogen-containing base. Let's look over here. Here's the base. That's the base. Look at it, has nitrogen in it. None of the other parts of the nucleotide will have nitrogen in it, so that's how you can tell it's the base. A nucleotide will have some sort of sugar, either ribose in RNA or deoxyribose in DNA. Here's the sugar right here. That's the sugar. And it will have a phosphate group. and they'll be attached in that order. The base will be attached to one side of the sugar will be in the middle, and the phosphate group will be on the other side. All right. So if this sugar is deoxyribose, this will be a DNA nucleotide, and this base could be adenine, cytosine, guanine, or thymine. If this is an RNA nucleotide, this sugar will be ribose, and this base will be adenine, cytosine, guanine, or uracil. We link nucleotides together to form a nucleic acid by dehydration synthesis. And instead of like the other ones you saw linking one right next to each other, we'll think about one on top of the other. An oxygen and a hydrogen from the phosphate group interacts via dehydration synthesis with an oxygen and a hydrogen from the other nucleotide. We lose water, and then we connect the two nucleotides with that phosphate group in between. Deoxyribose sugar in DNA, phosphate group, next sugar. This part here will become very important later when we talk about genetics and DNA replication. Remember how in carbohydrates we numbered the carbons? So we find the carbohydrate, the sugar. Remember we found the oxygen and we go number clockwise. 1, 2, 3, 4, 5, 6. Right? Remember that's what we did? New nucleotides are always added to the 3' end of the polymer chain. So if this is our polymer, we're adding our new nucleotide to this 3' end. Adding the new nucleotide to the 3' end. And then this bond that occurs between the two sugars in a nucleic acid with the phosphate in between is called a phosphodiester bond. So this right here, phosphodiester bonds. Phosphodiester bonds. DNA is also, antiparallel. What that means is that the two strands of DNA run in opposite directions. Okay, so if I had nucleotides oriented, this is going to be a really bad picture, but I'm going to do it anyway. Oh yeah, this is not going to be good. Draw. And then here's the base. Oh, what am I doing? There's a picture right here. Oh, geez. All right. Well, you know what? We'll just use this picture. Thank God I didn't have to draw any longer. They're anti-parallel, which means if I look at this side, this strand, and I number the carbons, 1, 2, 3, 4, 5. The 5 carbon of each sugar is pointing this way. We call that the 5 end. 1, 2, 3. The 3 carbon of each sugar is pointing this way. It's called the 3 end. If we go on the other strand, whoa, it's upside down. What are we going to do? We're going to start the oxygen. We're going to count clockwise. 1, 2, 3, 4, 5. Now look at the 5 end is towards the bottom. The 3 end is towards the top. That's what we call antiparallel. One side is flipped over from the other. Our bases pair together via hydrogen bonds, all right? And adenine and thymine always pair together, and cytosine and guanine always pair together in DNA. Adenine and thymine will always be found in a pair. You'll never find adenine and guanine or cytosine and adenine. Adenine and thymine, cytosine and guanine. If this was in RNA, well, where there isn't a T, we would have uracil in the spot for T. All right? But that's basically it that I wanted to mention that you need to know about DNA, RNA, and nucleic acids, about their structures, properties, and functions. All right? All right, if you have any questions, let me know. Otherwise, we'll go over this more in our live Zoom session.